first part of the plasmid prep involves the harvesting of the E. coli cells. So I have this flask of culture here, and what I have here are some microfuge tubes. So these are 1.5 milliliters or 1,500 microliters. So what I'm going to do is basically put the E. coli culture into these microfuge tubes and fill it most of the way to the top. You got to leave a little bit of room in the lid for a tiny bit of air so that the lid goes on properly, but basically I'm going to fill them most of the way to the top, and then they are going to go into the microfuge. Here's the microfuge. Basically it is a small centrifuge that is custom designed for these microfuge tubes. So if you take a look, I can program the settings. Right now it is set for 1 minute and 15,000 revolutions per minute, 15,000 RPM. Basically what you need to do is take off the lid. I'm going to put in 2 tubes, and they're going to go in opposite one another. It's very important when you're centrifuging that you do balance the centrifuge. This lid is going to go on. I'm going to fasten it so it's finger tight, and then we are going to hit start. The microfuge just finished and the lid popped open. Let's take a look at our samples. Now if you take a look very carefully, there's a small amount of E. coli in that test tube. So what I'm going to do is pour off the liquid supernatant, and I'm going to basically repeat the procedure in order to try to get a larger pellet. And I'll show you what that pellet looks like at the end of that procedure. I repeated the first part of the procedure a few times, so I added more culture, spun it down, so I think I've done it about 5 times. And let's take a look and see if we have a larger pellet now. And yeah, you can see that's a lot better. We have a larger pellet of E. coli. So what I'm going to do now is basically remove the liquid supernatant before proceeding to the next step. So at this stage, what I'm going to do is resuspend my cells in this PD1 buffer. So the PD1 buffer is an isotonic solution, and it also contains RNAs, so it's going to digest away any RNA that's found in the solution. I'm going to use this micropipetter. So this micropipetter is a device for measuring small amounts of liquids, and I'm going to measure 200 microliters inside the micropipetter tip. So here goes. The way I'm going to do this is basically get the solution. You can see that's what 200 microliters looks like. I'm going to resuspend the cells, and the way I'm going to do that is basically by pipetting up and down until the cells are in solution. I'm going to continue to do this, and then I will proceed on to the next step. In the next step, I'm going to add 200 microliters of PD2 buffer. So PD2 buffer contains a couple of ingredients. The first ingredient it has is a detergent called SDS. SDS stands for sodium dodecyl sulfate. So what the sodium dodecyl sulfate does is it's a very strong detergent, and what it will do is actually dissolve the membranes of the bacterial cells. So it's going to dissolve those membranes, and it's going to lyse the cells. They're going to break open. You're going to have all of the contents of the cells will come out. The other ingredient of buffer PD2 is it has sodium hydroxide in it, so it's alkaline and quite highly alkaline. And so what the alkaline solution is going to do is denature proteins, and it's going to denature DNA. So hydrogen bonds between the 2 strands of DNA are going to come apart. This is mostly going to affect the genomic DNA because the genomic DNA is not supercoiled. So it's going to get all precipitated and tangly, and that's going to allow it to come out of solution. The plasmid DNA is supercoiled, so it's not going to be affected as harshly and should stay in solution at this time. I leave this and let it sit and lyse the cells and denature the proteins and genomic DNA for 5 minutes. So what I'm going to do now is add the PD3 buffer. And the PD3 buffer is an acidic sodium acetate solution. So what that is going to do is neutralize the sodium hydroxide from the previous solution, and then that's going to allow the DNA to form new hydrogen bonds, but this time it's going to be all tangled and insoluble. but the plasmid, like I said, will remain soluble because of its supercoiling. So if you watch here, I'm going to add the PD3 buffer, and you should see the precipitation of the DNA and the proteins start to happen right away, although I am going to incubate this on ice for 5 minutes. So there we go. I'm just going to mix it up a little bit. Maybe you can see it's already getting a little bit cloudy, and that's the precipitation of the proteins and the genomic DNA. So like I said, I'm going to let that incubate on ice for 5 minutes. So there's the tube after it's been incubated for 5 minutes. You can see all the precipitate in there. So the next step is to put this in the microfuge for 5 minutes and get the big white pellet in the bottom. So like I said before, the white pellet is going to be your genomic DNA and all sorts of precipitated proteins, and this plasmid and other solutes will remain in solution. Our 5-minute spin is up. Let's take a look at the precipitate. So there it is. Hopefully you can see it. You can see the precipitate has come out of solution. So what I'm going to do now is use one of these little spin columns. So this is the technology that comes with the kit. That little white filter at the bottom is a special glass filter that is designed to bind DNA. So at this point, the liquid that I have in my tube from the previous step is going to contain anything that's soluble. So we're looking at all sorts of electrolytes in the cell, maybe glucose, all sorts of things like that. And of course the plasmid DNA. So I'm going to apply this liquid here to this column, and then hopefully that will bind the plasmid DNA. So very carefully I'm going to get that liquid out, and none of the precipitate is going to be added to this column here. It may take me a couple of rounds of pipetting. This is also going to go in a collection tube, by the way. That's going to collect any liquid that goes through. Very carefully. Like I said, I don't want to get any of the precipitate. The precipitate is going to have contaminating proteins and genomic DNA. These are things that we do not want. So that's done. So this here now is waste. I'm going to throw that out. I'm going to keep this. You can see this is has a nice little lid. That's going to go in the centrifuge. It's going to get spun. The liquid is going to go from the top to the bottom. As it passes through the filter, the DNA in the plasmids will be captured on the glass filter. Here it is after the centrifugation. You can see the liquid in the bottom. Like I said, this is going to be waste, so I'm going to dispose of that. I'm going to put the spin column back in the collection tube, and the next step is I'm going to wash it. So over here I have a wash solution, which is mostly just ethanol, and ethanol is a good wash solution for DNA because it will not react with it, and it evaporates very easily. So 600 microliters of that, and this is going to go back into the microfuge tube to allow the ethanol to pass through the filter and wash the DNA. So this has gone through the centrifuge. You can see that the ethanol has gone through the filter and washed this. So I'm going to dump that out. This is going to go in the centrifuge one more time, just as a drying step to get out any excess liquid or ethanol. This is out of the centrifuge now. I'm done with the spin column. That's going to get thrown out. And at this stage, what I'm going to do is now put this into a new centrifuge tube, and this centrifuge tube is going to be used to collect the DNA. So I'm going to open this. The spin column can go in here, and I'm going to add something called elution buffer. So the elution buffer is a higher pH, I think it's about pH 8 solution, and what that does is allows the DNA to unbind from the glass matrix and be collected in the bottom of the tube. So for this step I need 60 microliters of elution buffer. I'm going to apply it directly to the column matrix and let that sit for 1 minute to soak in before I centrifuge. I've done putting this through the microcentrifuge. I'm going to remove the column. I'm done with the spin column now. Close the lid. You can see what I've done is I've labeled this for the name of the plasmid, which is P Biology 107, and there's the plasmid in the bottom. That little bit of liquid there, that is the plasmid DNA, and it is ready now for the gel electrophoresis and also for the lab next week.